Good afternoon, everybody, or good evening, good morning, depending where you are in the world. I see there is people from all around the world, from Portugal, from the UK, and also in California, in the United States. So welcome, everybody. We are going to have a webinar about the soil microbiome as a tool to improve nutrient management. And I hope this hour that we will spend together is really interesting for you. I'm Pablo Nebreda. I'm the head of marketing for BiomeMakers, which is the company that we are hosting this webinar. BiomeMakers is a smart microbial discovery startup based in Sacramento, in California. And we connect soil biology into decision-making processes in agriculture to benefit farmers and reverse the degradation of arable soil, encouraging carbon sequestration in soil too. Through our leading platform technology called Becrop, we measure the biological quality of the soil and deliver agronomic insights to optimize farm operations. We will talk about this in a minute. So I will present Francisco López, also named Paco, from our team, which is one of our agronomists, and he will deep dive more in this topic. And then after that, we will have Brian O'Toole and Rod Carlsen that are joining also this webinar from TerraVesco. TerraVesco works hand in hand with the genius of nature to feed the soil, growing healthier plants and more nutritious crops organically and sustainably. TerraVesco produces professional-grade OMIR list and CDFA certified organic berry compost and vermi extract. So welcome everybody, and I'm gonna ask Paco to join here and start this part of the presentation. Before that, just to mention that you have below in the lower right part of your screen, you have the ability to ask questions, and we will hold a Q&A session after the two presentations. So it will be around 20 minutes of Paco, and then another 20 minutes for TerraVesco. And after that, we will have all the Q&A. So feel free to ask questions as we go, and then we will cover that later on. Thank you, everybody.
Okay, well, thanks everybody for joining us. I'm Francisco López, Paco. Thanks for the introduction, Pablo. I'm an agronomist from Spain, based in California now. Most of my experience is in viticulture and the wine industry. And since a year ago, I'm the lead agronomist here in BiomeMakers. So I'm happy to tell you a little bit more about not only our company and the services that we offer, but also some general information about the microbiome based on the scientific literature that I think will be super interesting to share with you guys. So let's jump into this. I hope everybody's seeing my screen all correctly now. Pablo, if you can please confirm.
Yes, the screen is loading. It should be live in a second.
Fantastic. Okay, let's get started then. Once again, thanks for joining us at this little webinar by BiomeMakers with our friends from TerraVesco in collaboration. And I hope you find this interesting and informative. This is sort of the agenda of today. We'll talk a little bit about the soil microbiome, soil health, who we are at BiomeMakers, then a little general information about the soil microbiome, especially involved with plant nutrition. And finally, we'll look at how we at BiomeMakers can help with our functional analysis, improve nutrient management and improving general soil management. So let's start by defining the soil microbiome, which is defined as the interacting community of microorganisms that inhabit the soil. And these are several types, from bacteria and archaea to all these different types of fungi and protozoa, which are also really important for nutrient cycling in the soil. Then once we talk about the soil microbiome, we'll probably think about the concept of soil health as well, which it's an interesting thing to mention because only living things can have health by definition, right? So in order to talk about soil health, you have to consider the soil a living organism. So the definition that USDA gives to soil health is the capacity of the soil to function as a living ecosystem that sustains life. Can also be defined as symbiotic ecosystem that can be managed and is managed to provide nutrients for plant growth. But the take-home message here is that soil is not an inert thing. It's not just dirt below our feet. It's living organisms like the microorganisms in it, and like animals and humans even. So what we want to do is be able to model soil functionality to provide data to enhance the productivity of all type of soils. Who are we with all these challenges ahead and all this stuff we're trying to do? Well, the BiomeMakers, we're a Silicon Valley startup that now has global presence and almost everywhere in the world, at least every continent except for Antarctica. However, maybe with soil, with climate change, we might be there soon. Functional analysis, and we are comprised of a qualified team that I'm going to introduce to you really fast. These are some of our genius in our team. We have a lot of very educated and well-regarded people in their own fields. Alberto Acedo, our CSO, is a PhD in genetic medicine that then jumped into the soil microbiome field, that has a lot of knowledge in the sequencing field, as you can see in all those prizes. Daniel Almonacid, another one of our geniuses, one of the most prolific inventors of patents in the microbiome field in the world, as much as all these other PhDs that you see in the picture, some other people with masters, and all kind of really fantastic and well-educated people that help us do what we do. More than 30 people divided in 2 labs, one in the USA in West Sacramento, California, and another one in Europe, concretely in Spain, in the city of Valladolid. We're backed and trusted by many investors, accelerators, as well as institutions, as you can see, including USDA and the European Commission, as much as many other entities. And most importantly, recognized by customers all around the world. As you can see, some familiar names there, like our friends from TerraVesco you'll be hearing from later on today. Let's go into the soil microbiome and its function in the nutrient management, nutrient cycling in the soil. I'd like to start with this. The soil microbiome that we already defined is obviously affected by many different factors, right? The first of which are going to be the temperature of the soil, the sunshine, which is going to obviously regulate this, and the rain. And these directly links with the soil characteristics, right? The percentage of organic matter is going to be very important, the pH as well, but also is the texture, because it's going to influence the availability of water or oxygen, which per se are some of the most important factors, and are directly linked to, as we were saying, temperature, rain, and so on, due to the water holding capacity and conductivity of the soil. And these would be just some of the factors in the soil, since there's many others. But the plants also have an influence, right, by the exudates that they introduce into the soil, which are going to be different based on the phenological stage of the plant, its nutrient needs, and the type of stress that the plant is, because it might work to gather microorganisms that might help or might supplement the plant with different beneficial molecules, as we'll see in a little bit. And obviously, humans play a role here, right, through farming management practices, such as obviously soil management. Till versus no-till has been proved in the literature to have a direct effect on the soil microbiome, as does the use of fertilizers, and of course the use of pesticides. So that's kind of gives us an idea of how dynamic and how affected by so many factors the soil microbiome is. Going into the field of plant nutrition specifically, most of you might already know about this. It's not super. It's kind of general information, but I think it would be a good reminder for some people who might not be familiar with this. And none of this is my work. I just did a little literature review of some fantastic scientific literature out there, and I will put the bibliography at the end in case anyone wants to go back to these references and look at them into more detail. But as you all know, soil microorganisms cycle many nutrients in the soil, right? And they metabolize all these different forms from organic forms into mainly inorganic forms that are going to be the ones that are soluble and that are plant available and that the plants can uptake from the soil solution. And I want to focus here on the main macro elements: nitrogen, phosphorus, potassium, and sulfur. In the case of nitrogen, one of the most abundant elements in the atmosphere, right? 80% of the atmosphere on Earth is elemental nitrogen, but plants can just take up nitrogen from the soil. So how does it end up in the soil and end up being existing in forms that the plant can use? Well, the plants prefer nitrates and ammonium, and which one they're going to use is going to depend on many factors, such as the type of plant, right? For example, blueberries, for example, tend to prefer ammonium, whereas grapevines would prefer nitrates. Then the type of soil, with especially the aeration and the pH, is going to influence the form that it's more prefer. And then there's many other factors, such as the cost that it has for the plant to use, for example, nitrates versus ammonium. But in short, these are the two types of forms that the plants can utilize. That's not elemental nitrogen, right? So how does it end up there? Well, there's many processes. One that it's very well known would be nitrogen fixation, that most of us are probably familiar with, and that consists in the fixation of elemental nitrogen in the soil into forms, in this case, ammonia, that can be then cycled and finally utilized by the plants. But that's not the only pathway, right? Nitrogen fixation, there's many other pathways in the nitrogen cycle where microbes are involved, such as the mineralization of organic nitrogen into ammonium, the nitrification of ammonium into nitrates, then denitrification, which might cause that loss of those nitrates out into the atmosphere, and many more, such as nitrate reduction, anaerobic ammonia oxidation. So it's a really complex cycle, fascinating to study, and most of it is driven by microorganisms in the soil, especially bacteria and archaea. In the case of phosphorus, well, again, plants can take any type of phosphorus, and they prefer orthophosphates, right? They just can't take organic phosphorus in humus or in the soil. So how do microorganisms help in this cycle to provide the plant with these orthophosphates? Either through phosphorus solubilization, by creating these organic acids that help dissolve minerals that contain organic phosphorus and are going to make this phosphorus in a form available for the plant, or through mineralization, similarly to what happens in nitrogen as a concept, right, where this organic phosphorus is transformed into inorganic forms that the plants can finally uptake. A really important player in this phosphorus cycle is the arbuscular mycorrhizae, which are fungi that live in a symbiosis with the plant in certain environments and with certain plants as well, by colonizing the root cortex, or the external layer of the roots. It's a symbiosis because they help the plant uptake a lot of different nutrients, especially phosphates, through enhancing the reach of the plant or creating with their hyphae a more extended net of roots that the plants can use. But in exchange, they take glucose or carbohydrates as energy from the plant, which can be up to really high percentage, up to 20 or 30%. They can even, these relationships, especially in cases where there's a lot of phosphate and the plants don't really need this mycorrhizae, can even not be beneficial. But in most cases, especially in conditions of scarcity of nutrients, especially phosphate, this is a symbiotic relation that is going to help, or through which microorganisms, fungi in this case, are going to help the plant not only uptake phosphorus and other elements such as potassium, sulfur, even nitrogen, and some micronutrients, but also increase the tolerance to different stresses such as drought, salinity, etc. So really interesting, the role of arbuscular mycorrhizae in the soil. As for potassium, it's really abundant in the soil, but most of it is immobilized into other minerals, and only a small percentage of it exists in the form of the potassium cation that the plants can use. So this is where microorganisms help with the role as potassium solubilizers by improving the availability of potassium and creating more of these potassium cation that the plants can uptake from the soil. Sorry. And below are some examples, since I think this is not as well known as the effect of, or the role of mycorrhizae as well, of some of the particular species that do this role, and then a really interesting paper where that figure was taken from as well. It's the way they work is, in simple terms, pretty similar to what happens with phosphorus. They produce organic acids that lower the pH, not in a huge amount that might be sensed through like a soil analysis, but in really particular parts where the potassium cation that's bound to some minerals is going to be released and made available for the plant. And there's some other studies recent I'd like to mention that hypothesize that these microorganisms can also mediate the transfer of potassium and induce plant genes that enhance this uptake. So it's another really interesting topic that's being studied at the moment. Finally, in the case of sulfur, this is not as well known, but the sulfur cycle is really complex, as much as the nitrogen one, and it's sort of similar to the nitrogen cycle in the sense that plants can uptake one form of inorganic sulfate, mineralization, redox processes that finally provide the plant with the forms that they can uptake. Another role of microorganisms in the soil, not directly linked to nutrients through cycling, but through other ways, is the role of plant growth promoting microorganisms that are pretty trendy, and there's a lot of talk about them, right? Especially with the market of biostimulants being so active lately. Basically, what these microorganisms can do when they exist in the rhizosphere is, as their name indicates, is promote the growth of the plant and the development of the plant by supplementation of phytohormones that the plant would produce by itself, but that when are produced by microorganisms in the soil and provided to the plant, basically prevent harmful effects of stress or help withstand stress situations of drought, salinity, even nutrient deficiency or heavy metal contamination. So this is another example of beneficial microorganisms that you want to have as friends in your soil to aid your plant under this stressful situations. And I like to recommend this paper, one of my favorites in this topic, by Gamberdieva and others, on 2017, that has its a literature review with a lot of information about this topic. So if you have time and are interested in this, please go ahead, look for it and give it a read. This is some of the references that I use for this very short literature review, and I also encourage you all to check them out and read more. And if you want to see some papers by the BiomeMakers team, I would recommend you to check Alberto Acedo, or CSO, his ResearchGate profile, for example, and see some of the papers where we have been involved, especially if you're interested in wine and vericulture as I am. There's some really interesting recent papers that I would definitely recommend you to check that are related to them, microbiome networks, and all these stuff. Now you're probably wondering how can you guys at BiomeMakers help nutrient management with your soil functional analysis? What's that soil functional analysis that you're talking about? So by using the technology that we use, which is called amplicon sequencing, we can characterize the soil microbiome and obtain information about different things, such as with our basic analysis, we get information about soil microbial biodiversity, pathogens that inhabit the soil for your specific crop, potential for nutrient cycling, as well as other stuff such as presence of biocontrol and plant growth promoting microorganisms. Then through the use of Geome, which we call the big brother of Becrop, seems like the picture or the logo unfortunately didn't work, but this is type of trial for testing the effect of inputs or testing the effect of different practices in the microbiome with statistical reliability that can be used to try or test the effect of different biofertilizers or compost or whatever product in the soil microbiome, check the duration of inoculated species in the soil and so on, and see if this product that you are applying is doing something good or bad for the soil, or this really promising biostimulant is actually working or not so much in your type of soil and crop. And finally, we can determine the microbial composition and quantify the microbial communities in biological products with what we call Becrop product. And some of the examples of things you could test with this would be your compost, biofertilizers, compost tea, biodynamic preparations, you name it. I'm sure we'll hear more about this from our friends at TerraVesco. And the way we do this is extracting all this DNA information from the soil, transforming it into biodata, and creating a functional analysis report, in that case, Becrop, that could also be used to do Geome or Becrop product. And as you know, this could be used for several applications. We provide specific solutions to specific problems, especially thanks to our great team. So we're working in projects involving yield improvements, checking crop quality and its relationship with the microbiome, testing app products, as I just defined, monitoring farm practices, checking or monitoring disease risk based on the abundance of pathogens. And in the case of wine, coffee, even cardamom, as I was talking with a client this morning, we can help with the microbial terroir characterization. How does this work? You provide a soil sample taken directly from the soil as a composite sample. The surface that you can cover with one of these samples is obviously going to depend on how homogeneous the soil is, based on those factors that we defined before, right? Especially texture, pH, quantity of organic matter, and obviously management are going to be important. But just imagine you're taking composite soil sample at around 6 inches deep, and you send it to our lab in soil Sacramento, in West Sacramento, using our shipping system and following our proprietary sampling methodology. With this, we're going to produce data. How do we do this? Well, we first isolate and purify the DNA that is in the soil, the information about the microorganisms in this soil. Then we put it through a PCR, polymerase chain reaction system, to basically amplify and duplicate several hundreds of times this DNA, so we have more quantity that we can analyze. And then we use what's called sequencing, high throughput sequencing, this modern technology that basically transforms DNA into letters or into data that we can analyze with bioinformatics. The third step is interpreting this data, right? We do this with proprietary on pipeline and databases using bioinformatics, machine learning, and this information that's based on 3 layers, right? First of taxonomy, then functionality, or what microbes are present, what are these microbes doing, or what are their functions, and lastly, network or ecology, how are they interacting? And at the end, we deliver this report. What you're seeing there is the first page of a Becrop report that some of you might already be familiar with, and it's crop specific, has all these parts, but it's basically divided into biosustainability, different takes on microbial biodiversity from a taxonomical, functional, and ecological standpoint. Then what we call health or healthiness, which is going to look at the detection of pathogens for your specific crop in that soil, as well as the detection of beneficial species, plant growth promoters, biocontrol agents, and so on. And finally, information about nutrient pathways, or based on the genes that we are detecting, what's the potential of this soil to be able to mineralize nitrogen, for example, and provide ammonium that the plant can uptake or that can be nitrified? Or what's the potential of this soil to solubilize phosphorus and provide the plants with orthophosphates based on its microbial content. That's kind of the information that we provide, and that can help know more about a certain type of soil, see which soils would benefit from a certain type of product, etc. And as I was saying, this is our goal for helping with nutrient cycling through this technology. We want to monitor soil functionality, so you know what's the status of, for example, your different blocks in your farm, which ones might be less biodiverse because they've been maybe mistreated in the past or intensively treated, and you want to pay more attention or be more gentle with them. And also provide information about where and when it would be the most beneficial to use certain inputs and practices to increase this microbial nutrient supply. Since these soils where the nutrient supply pathways are potentially low are likely to benefit the most from these applications, right? Compared to a really active and a really healthy soil. And finally, this would result in a reduction in the cost of fertilizers, especially in organic fertilizers, which in many cases are overapplied, and eventually an improvement in soil health, which is what we all want, right? Since better soil, it's going to provide better food, and it's going to also sequester carbon, which is so important for climate change, etc., etc. And the way we just, and I'll be brief since my time is coming to an end, is through these three main services. Becrop, which is our soil functional analysis, a one-time single report. Then Becrop product, which is an analysis for products such as compost, as I said, biodynamic preparations, biofertilizer, and analysis that uses the same technology, but it's applied to a product itself. And finally, with Geom, as I defined it before, the bigger brother of Becrop, which is a testing protocol for inputs or management practices that is has a more comprehensive experimental design, more complex, but that provides a statistically reliable data that can be used to write a publication, justify your claims, etc. So I'll really briefly describe the three main services that we use. Just please keep in mind that the technology behind them is all based on amplicon sequencing, as I defined, and that, of course, if you're interested in any of this, send me an email, send us an email, check our website. We'd be happy to help you and see how we could apply this to your farm, to your management, or to your product manufacturing. So the first one would be Becrop, the one-time functional analysis based on a composite sample of soil that, as we said, it's created from these biodata extracted from the soil into all-in-one report that has these three main parts: biosustainability, microbial biodiversity of fungi, bacteria, and archaea, health as detection of pathogens for your specific crop, of course, and detection of beneficial species, biocontrol agents, plant growth promoters, and other microorganisms which can produce molecules that help the plant overcome different stresses. And these nutrition potential based on the detection of genes that are going to tell us the potential for these microorganisms to do these different pathways or produce these different enzymes that are going to cycle nutrients. I think I'm going to skip that since we don't have much time, and I'll go directly into Geom, as I was mentioning, the bigger brother of Becrop, which it's type of trial used to test the effect of different inputs such as biofertilizers, compost, or normal fertilizers, you name it, in the soil microbiome. And that I call it the bigger brother because it's comprised of several Becrop analyses. The way we do this is we ask the person that wants to do this study, or the team that wants to do this study, to select a control zone and a treated zone to be studied. The control will be untreated, and in the treated, let's say you want to try the, the inoculation with trichodermas. And then we are going to test these soils several time points before the start of the study to have an idea of how the soil were before we start doing anything to them. And then after, let's say we do this for trichodermas, maybe 15 days later, a month later, and then 3 months later, to see how the evolution, first the abundance of the specific microorganisms in study are changing, and how the evolution of these pathways that they might be changing are evolving as well. And most importantly, we want to do this ideally in 3 or more different locations to have information about, for example, a sandy soil versus a sandy loam, and then a clay soil. And last but not least, 3 replicates per time point. 3 replicates mean that in each one of these time points in the control zone and in the treated zone, we're going to repeat the analysis 3 times. What is this for? Isn't this redundant? No, this is for being able to run statistics that are going to give us information that we can know is reliable. So as you can see, at least triplicates, 3 analysis per time point, and treatment and location. This treated and untreated zone, at least 2, but ideally more time points, and ideally 3 geographical locations. What are the results that we get? Well, we get the percent change that can be attributed to the product use or to your management practice or whatever you want to test, or your hypothesis, over certain time periods. And most importantly, the statistical significance of these changes. In other words, whether you can be sure that these changes were due to the product or to a management practice, or if you can really be sure that this is a product and it might as well be chance. So in short, trial study with a complex experimental design to test the effect of practices or products in the soil microbiome and get statistically robust conclusions that are going to allow us to justify our claims about our products, or choose a product that actually work if you are the grower, or even in the case of manufacturers, discover new modes of actions that you didn't know about these specific fungi, bacteria, or whatever you're applying into the soil. Lastly, as I said, we offer Becrop product, which is used to verify the composition of biofertilizer, compost, any biological input. And the way this works is it's based on our same sequencing pipeline, but instead of soil, our clients send us a sample of compost, for example, or any biological input. So we analyze it in triplicate, and we give information about the potential strengths of this product in the microbiome by studying the microorganisms in the product and what are the potential functions. And then we identify all the bacteria in archaea, all the fungi in this product, and we tell you the absolute number of bacteria in colony forming units per milliliter or per gram in your product, and the relative quantification of fungi, or the percentage of the different fungi that comprise your product, which would be like something like this for the absolute quantification of bacteria, and like this for the relative quantification of fungi. Again, this is Becrop product, an analysis not for the soil, but for your compost, your biodynamic preparation, you name it. I'm going to finish with this. Thank you very much, but I didn't want to leave without mentioning that at Fields Forever, we are offering this global soil health initiative that we call Fields Forever, through which we are providing free and or discounted Becrop analysis for growers, manufacturers, and especially for researchers who want to work more in the field of soil health and want to jump into, want to get better research to help us know more about the soil health. So if you're interested, you want to maybe try Becrop in one of your farms, or you're a research from a university or a private entity and want to utilize these analysis for your projects, please check fieldsforever.com or check our website. And the second open call is open, so please hurry up, because there's not that many free samples left since the first one was really successful. This would be it for my part. Thank you very much, everybody, for your patience and for listening to me for 20 minutes or more. And I'll be happy to answer any questions after we hear from our friends in TerraVesco. So yeah, thank you. And
thank you, Paco.
Thank you very much, Pablo.
So thanks for this interesting presentation. In the interest of time, I'm going to call our friends of TerraVesco to join us here in the live conversation. They will speak about, they will present their products, and also in relation with a vineyard case study, in which they're gonna use our Becrop technology to evaluate the impact of vermi extract and see the effects on soil health, yield, wine quality, and return on investment on that particular project. So let's wait for a second for them to join.
I can see myself.
Hello. Hi, Brian. How are you doing?
Okay, I can't see you, but I'm going to take you through the slides. This is Brian O'Toole of TerraVesco. We certainly appreciate the opportunity to participate in the BiomeMakers presentation today. We are a client of BiomeMakers, and we work extensively with them in our growers and farmer customers to have a better understanding of soil microbiology and fertility. And we think that the Becrop report is a very important tool in helping our clients better understand their soil health and fertility. We're a company located in Sonoma, California. We were founded back in 1992. We are a producer of microbiology, and our core products, vermicompost and vermi-extract. Our products are certified organic with OMRI and CDFA. And being here in the wine country, our business really started primarily in the wine grape and vineyard side of agriculture, and it over the years has expanded into tree nuts, berries, cannabis, hemp, and landscaping. Just real briefly to give you an idea of what our production process looks like. We take organic dairy manure that we collect from dairies throughout Northern California. That material is put through a thermophilic composting process, which eliminates weed seeds and any harmful pathogens. We then feed that pre-composted material today to more than 80 million red wiggler worms. We have a completely natural production process, and then ultimately it creates our core product called vermicompost, which is then fed to plants and it helps improve soil health and fertility. When we talk to farmers and growers about soil health, there's lots of confusion out there. Lots of farmers and growers express that they've had a lot of frustration in trying to read soil tests and understand how that information gives them a perspective on soil health. And one of the concepts that we've used very successfully over the years, we call the soil health pyramid. And what the soil health pyramid does is it breaks soil health down into three basic fundamentals: composition at the base, nutrients in the middle, and what we believe is the new frontier of modern agriculture, which is the microbiology. Making sure that we have a clear understanding of the beneficial microbes in the soil and how those contribute to soil performance, soil health and plant performance. Next slide. There are basic two methods of sourcing microbiology. Paco has talked about some of those. Biostimulants. There is the genius of man approach, where we have bioengineering that takes place and produces limited populations of bacteria and or fungi to improve plant performance. In our business, we are leveraging the genius of nature. We have had our product analyzed for bacteria and fungi. We have witnessed over 2,700 species of bacteria and over 300 species of fungi in our products. And we think that's an intelligent way of introducing or colonizing soils with the kind of beneficial microbiology that can improve soil health and performance. Our base products, again, are vermicompost TerraThrive Pro and TerraThrive Pro vermi-extract. And at this point, what I'd like to do is to introduce Rod Carlsen in our organization. Rod works in the trenches and in the fields every day with our growers and farmers, and we thought it would be most effective to see an actual case study of how we've worked successfully with the folks at BiomeMakers using their Becrop reports and Geome reports to help a wine grape grower and vineyard manager produce better results in the vineyard. Rod?
Okay. Thank you, Brian, and thank you to Paco and Pablo for extending the invitation to participate in this webinar, and thank you to all over the world that are participating in the webinar. So Winery in northern Sonoma County, Francis Ford Coppola, you might know the Godfather films, approached us back in winter of 2019 about evaluating our liquid product, the TerraVesco Pro Vermi Extract, as a way to mitigate some hillside blocks that go into one of their estate wines called the Archimedes Cabernet Sauvignon from Alexander Valley. And the winery is very happy with the quality, but it's prone to berry dehydration late in the growing season. So Coppola wanted to evaluate how the addition of our liquid product via the drip irrigation system could help to mitigate the dehydration in the fruit close to harvest. So it's same clone of Cabernet 337 on 2 different rootstocks, 140R and St. George. You can see the fertility programs that were followed in 2019, 2020. The Vermi extract regimen was identical. So both in 19 and 20, it was 2 applications, pre-bloom and berry set at 20 gallons to the acre per application. With respect to added nitrogen, they added 18 pounds to the acre of urea-based nitrogen in 19, mid-season, sort of post-bloom, and then again early onset of ripening or veraison. In 2020, interestingly enough, the second year of the research, they were able to back off and cut their applied nitrogen by 44%, instead opting to put out 8 pounds to the acre of urea nitrogen pre-bloom prior to the first application of Vermi extract. In terms of investment for Vermi extract, looking at $216 an acre. The blocks were harvested in 2019, early October, a little bit earlier this year, last week of September, and the yields were separated and recorded by treatment. Special shout out to Redwood Empire Vineyard Management for that. And then in terms of the 2 treatments, they were crushed and fermented separately. So kudos to Coppola for pulling that off. And then it should be noted that 2019 and 2020 were markedly different vintages. 2019 was a pretty even-keeled, slow, gradual ripening. The area did have wildfire, but it showed up well after this fruit was picked, thankfully. But this year we did have wildfire that appeared in mid-August, and a lot of smoky days that hung around through September and definitely impacted the fruit, along with a lot of high heat events as well. And that's reflected in the yield comparison. So in 2019, just a more benign set of growing conditions, and we saw a healthier crop across the board. But in year 1, the Vermi extract treatment increased yield by 7.6%, which we were thrilled to see. In 2020, because of the myriad of challenges related to fire and heat, prolonged heat events and smoke-filled skies, the winery actually had to conduct some small batch fermentations to decide whether or not they were going to harvest the fruit, and thankfully they did. And we're very proud with that 4.5% increase in 2020, given the myriad of challenges at hand. In terms of the wine from 2019, Coppola was kind enough to share the fermentation logs. I don't want to get too much in the weeds here, as wine is inherently subjective. But if you really focus your attention on the far right column, which is the total phenolics, Coppola and a lot of wineries for that matter are really trying to hone in on the combination of taste, color, and mouthfeel, or the perception of tannin with a darker colored wine on your taste buds or your palate. So using their software from WineCloud, we were able to improve that total phenolic composition of the wine with the 2019 vintage. Also very exciting for us. In terms of 2019 trends and 2020 calls for further analysis, this is when BiomeMakers comes in. So we carried out a pre-harvest soil sample through Oregon State's agronomy lab. We saw some positive trends in the form of increased active carbon, or the fully broken down organic form that really is the lifeline or the food source for microbial activity and function in the root zone. And we also saw increased soil moisture in both the Vermi extract treatments versus control, a positive indicator for the mitigation of the dehydration that they've been experiencing over the years in the fruit, and also contributing to the resultant yield increases that were documented. So a couple of questions that came out in 2019 were, number 1, what about fungal to bacteria ratio? And then number 2, the role of plant growth regulators or phytohormones in mitigating the effects of abiotic stress in the form of berry dehydration. So drum roll, BiomeMakers, we started talking with them winter of 2020, and we went ahead and did a Becrop analysis in this vineyard 12 days after the second application of Vermi extract in the latter part of June. They carried out their analysis and gave us back the following information, and in particular, the fungi to bacteria ratio is what I want to focus on. So in block 2, we moved it from 0.63 to 0.75. And in block 3, moved it from 0.65 to 0.70 in the areas treated with vermi-extract. You know, in the arena or the sphere of regenerative agriculture, there's a lot of discussion about healthy, productive biological soils being in sort of that 1-to-1 fungi to bacteria ratio. Most of the research, though, that I've borne witness to, typically they're talking about no-till annual cropping systems, or certainly reduced tillage annual cropping systems. Here we're in a vineyard, we're in a perennial system. There is a cover crop sown every fall, and the winery will come back the following spring, late in spring, and disc in and cultivate every other row, and then they'll alternate that with mowing the cover crop, really just to try to keep the dust down. And then the vine rows themselves are managed for weeds. So I would consider this to be a moderately intensively managed system. So for us to bump the fungi to bacteria ratio is very encouraging. Some additional interpretations and insights from the Becrop report, as Paco alluded to, the biosustainability, sort of the combination of what the addition of our product is doing to enhance the biodiversity, the functionality of those microbes in the soil interacting with what's already there, and then certainly resistance markers for the potential for pest control. With respect to nutrient pathways, also germane to what Paco was covering earlier, nitrogen use efficiency, there's a tangible reduction in applied nitrogen there, going down from 18 pounds of urea in 19 down to 8 pounds in 2020. Again, sort of the capability of a product like ours to mineralize organic nitrogen in the soil and even convert ammoniated nitrogen. In this case, the urea travels in a blend that contains the ammoniated form as well. With respect to phosphorus, as Paco alluded to as well, we saw the nutritional pathways for phosphorus being immobilized, improved in the vermi-extract treatments versus control. And this is really by function of the microbial mineralization of soil organic matter and making that phosphorus in the form of phosphate that the plant can access. With respect to potassium, microbial activity as well, but then also we have a humic acid fraction in our product, which is the fully broken down form of organic matter with the negative charge, and is really instrumental in helping us to kick critical positively charged cations like potassium off the exchange site and into the root zone and ultimately the vine. And then last but not least, the sulfur cycle too, in particular in block 2, we saw a dramatic opening of the pathway there, indicating that the microbes are helping to expedite enzymatic activity and also the sulfur cycle. With respect to plant growth regulators or phytohormones, when I got that Becrop test result back, I was pretty excited to get to this portion of it, and I fired a note off to Paco. In particular, we noticed very high levels of salicylic acid in the vermi-extract treatments versus low readings in block 2 control and then high reading in block 3 control. So I wanted to know more about that, and I'm not gonna, I give credit where credit's due, but basically Paco was insinuating that either by adding the product, we were supplementing the production of the compounds that produce that phytohormone already, or we have microbes in our product that are further enhancing the production of that compound. So I went back to the Becrop product report on our vermi-extract that BiomeMakers carried out earlier in the spring, and in fact, we contain one of the species that's known to aid in the production of the compound that produces salicylic acid, and that is Chromobacter xylosoxidans, which I'm not going to say 10 times. But it was interesting to find that, and then also find it boosted in the soils that were treated with vermi-extract. And why is that important? Well, it's important because in our region, rainfall can fluctuate quite a bit from year to year, and most of our rainfall falls in the winter and early spring months. And 2020 was a very dry rain year in Northern California. So the vines started off in a stressful position to begin with, and they're totally, in any year, even if we have high rainfall, they're totally relying upon late in the season, any water that's applied is going through the drip irrigation system. And wineries that are striving for high quality wine tightly regulate that applied water, as they don't want to overdo it and impact negatively the wine quality. So if you look at the drought conditions, you look at high heat, you look at fire, you look at extended exposure to smoke-filled skies, the fact that we were able to retain more moisture in the soil and also in the fruit with the help of boosting salicylic acid production, it's a compelling story. So to keep things consistent, in 2020, we also did another pre-harvest soil sample with Oregon State, saw a continuance of the same trends with respect to increasing active carbon, really that lifeline, that food source that the microbes depend on to carry out their work. And then we saw again increased soil moisture. Once again, these were taken mid-September, right before they picked. And then last but not least, that hormonal phytohormonal component that BiomeMakers was really able to help us delve into further and get a better understanding of what's going on. And then truly where the rubber meets the road is the return on investment to the vineyard owners. So in 2019, with that yield increase, we saw a return on investment of 225% on the use of our VermiExtract product. In 2020, we still saw a positive return at 24%. And given the myriad of challenges that we had to contend with, we're very proud of that result. And thank you. That concludes my presentation. And hopefully we still have some time for questions.
Thank you, Rod, and thank you, Brian, for joining us. I will call also Paco to join the screen, so we can go with the questions.
Here we are.
Hello.
There we go. Can you hear us, Pablo?
Sure. Yes.
Okay.
Yeah, everything was working properly. No worries. So Paco will join us also in a minute. There he goes. Hi, Paco.
There.
I just, yeah, now you have your microphone on. So we have 3 questions for you. We have, about 5 to 10 minutes. Try to answer all of those. So the first question is for Paco. It says, besides separating live and dead cells, how do you assess that microbial functions are active? Yes.
Okay, so first there was a question about whether or do we have the capability to discern dead from alive microorganisms? Mauro asked that. That's a great question, and for that we have validated the use of the PMA or Propidium monoacide, which basically what it does is before the PCR step, which amplifies the DNA, it binds to all the extracellular DNA or all the DNA that has escaped the inside of a cell. So it corresponds to a dead cell. So that way we can only amplify the stuff that it's alive and that corresponds to a viable microorganism. This is especially done for and requested for the analysis of biological products, right? When people want us to analyze their biofertilizer or compost, they want to see or they want to know what's alive in it. So that's how we do it. As for the interpretation of these functions based on the genes, well, for the functions such as biocontrol, plant growth promoting, and so on, those are going to be based on the descriptions in the peer-reviewed literature of the functions of these microorganisms in different soils and under different conditions and crops. For the nutrient cycles, we infer, as I said, the what genes are present, and based on proprietary and public databases as well, what is the enzymes that these genes are supposed to encode for, and in thanks to that information, give an assessment of the potential for nutrient cycling. This is though based on metabolomics, right? Not in transcriptomics. So we can tell whether the gene is there and the potential that it has to do these certain things. But at the moment, we are not assessing whether this gene is actually encoding or is transcribing for something. That would be the next step, which we hope to get to in a few years, would be the use of transcriptomics information. However, at the moment, the databases for transcriptomics in general for the scientific community are not very well developed. That's why we're sticking to the stuff we do with amplicon sequencing, and that's what we call potential for nutrient cycling. And so we can go into that next step of doing the transcriptomics and seeing the actual encoding of that enzyme.
Thank you, Paco. So we have two additional questions. Let's see if at TerraVesco they can hear us. Hello, can you hear us? No, let's try to put the question here in the chat. So there is a question in the chat that says, are those products from TerraVesco being used successfully on sports fields for turf? Are real examples of turf? Do you sell them in Europe?
We have conducted some research with Purdue University here in the United States on turf with some encouraging results. So we'd be happy to follow up and talk further about that.
Yeah, I think. Yeah, again, we've done some research in the past, and we've had some positive results working with turf, and we'd be more than happy to share those results with anybody that's on the call today that would be interested in that.
Okay, thank you. Okay, so thank you both Paco and Brian and Rod from TerraVesco.
Are there any other questions that you want to type in to us since we can't hear you?
Yeah, so let's go for the last question that we have here in the chat. So there it goes. That's the other question that we have. So do you have an idea of how much of soil health is due to the addition of microorganisms present in vermin extract with respect to the improvement of
We've just been tracking the differences relative to treated and untreated. I mean, through their sequencing, we certainly have the information related to what is native to the vineyard soil versus what changed when we added it. But that's the extent of where we are with that.
I mean, in essence, our products are colonizing soil with abundant beneficial microbiology, and that's where we're seeing the efficacy. At one time, we did some studies where they fumigated soils to kill nematodes before planting certain crops, and when they introduced vermicompost and extract into that process, we saw a recolonization of the microbiology in the soil that was lost during the fumigation process. So we know that our products do a lot to recolonize or colonize soils with beneficial microbiology.
Yeah. Well put.
So thank you. Thank you, Rod and Brian from TerraVesco, and thank you, Paco, also for your participation on this webinar.
I don't see any other questions for us. Do you?
Yeah, no additional questions. Also, it's just 1 hour past.
Yes.
So, as I said, thank you for participating, and we're gonna leave it here. So, just a reminder that this is gonna.
Thank you, guys.
Thank you. Glad you can hear us.
Paco and Pablo, we really appreciate the opportunity to participate with you today. I think it was a very informative presentation, and we'll look forward to doing these in the future as necessary. So, best to everybody who's listened in today. Thank you, guys.
Thank you. Bye. Have a nice
Thank you
day,
very much. Have a
or
good
night
one,
wherever
everybody.
you are in the world. Thank you.